Up until the late 1900s, fossil fuels were the main source of energy used on planet Earth. However, with climate change and global warming, we have seen the major consequences to this. Fossil fuels have now been deemed as an unsustainable source of energy, leading scientists to look for renewable and cleaner solutions for the future. Recently, scientists have looked to cyanobacteria and have made attempts of enhancing the process of photosynthesis as a key to a cleaner way of life. Photosynthesis is the process in which plants convert light energy to chemical energy. Light is captured by the antenna pigments, which get excited and pass this excitation onto the neighboring molecules. This happens until the excitation gets to a structure called the center of reaction. The center of reaction contains two chlorophylls that lose electrons, which will enter the electron transfer chain to create ATP energy. One water molecule is split by the light's action into oxygen and hydrogen, and the energy obtained from this hydrolysis brings the chlorophylls back to their reduced state. In this image, you can see the interior of a cyanobacteria. This organism uses an oxygenic photosynthesis method to create energy. What is special about the cyanobacteria is an additional structure that absorbs light much more efficiently than plants do. This protein is called the phycobilisome. This complex allows the absorption of wavelengths from the sun and is done much more efficiently than chlorophyll does in plants, making cyanobacteria a lot more effective. The phycobilisome is composed of two components called core and rods. These rods are constructed of the heterodimers of alpha and 2 beta subunits. The subunits form trimetric structures to make up the basic unit of the rods. The trimers then form hexamers to form the cylinder rods. There are two types of rods, which are the phycocyanin and phycourethrin. The phycourethrin absorbs green blue light and is located in the rod. The phycocyanin absorbs more red light and is located proximal to the core. Lastly, the linker proteins attach these rods to the core of the phycobilisome, which is made up of the allophycocyanin. Allophycocyanin is able to absorb the red light, which is the lowest energy level. As light hits the outer disk of the phycobilisome complex, energy is transferred down the disks towards the APC core. Light hits the outer disk first, and the energy is transferred by conformational changes. Once it reaches the core, energy will then be transferred into photosystem 2 through the linker proteins. In conclusion, by incorporating phycobilisomes into organic batteries, it is possible to increase the battery's efficiency and simultaneously produce electricity and oxygen.